Yes, thank you very much for the introduction and the opportunity to present UPyTher to you. UPyTher is a company that is entirely committed to developing medicines targeting the intraperitoneal drug space. So the intraperitoneal drug space, or the peritoneal cavity, is a confined space in your abdomen. It's formed by the peritoneum that lines the abdominal wall and the visceral organs. And it has proven to be an extremely challenging cavity in your body to target with drugs, because this peritoneum functions like a physical barrier that prevents the systemic drugs from entering the blood into your abdominal cavity, leading to an exposure problem. This also provides an opportunity for localized treatment. However, if you use existing systemic therapeutic products directly inside the abdominal cavity, they are rapidly washed out. So again, you have an exposure problem. So without proper drug products available, what you see nowadays is that clinicians are left with complex procedures to go in, associated with high morbidity and very poor outcomes for these patients. And this is in particularly a problem for patients with a condition that is called peritoneal carcinomatosis or peritoneal cancer. It's hallmarked by the spread of numerous tumors throughout the entire peritoneal cavity. And it's a major complication in some of the most common types of cancer, such as colon cancer, ovarian cancer, and gastric cancer. And because no good treatment is available, this complication provides the poorest prognosis for these patients, often with less than a year's survival ahead for them. Now imagine if you have a drug product available that does optimize this local exposure to your therapeutic, then you can change these patients from untreatable into a treatable group of patients. And that's what we are after with UPyTher. We are developing a completely first of its kind class of intra-abdominal medicines, and we do this based on our own proprietary polymer chemistry platform that provides a unique set of combination of properties in a material that you all need to overcome the challenges of intraperitoneal therapy. That means that our medicines behave like a liquid at room temperature, so they allow for convenient application, injection, spraying, you name it. And then after administration, they diffuse throughout the entire peritoneal cavity, and on site they transform from a liquid into a gel depot. And this unique combination of properties, that's also what sets us apart from existing gel systems and makes us superior for the use in the complex, hard to target body cavities such as the peritoneal cavity. So just before lunch, still going to show it to you. This is how it works in practice. This is a clinically relevant mini pig model. You go in there with standard laparoscopic procedure, no need for special equipment. As you can see, it behaves like a viscous liquid that you can spread throughout the peritoneal cavity. And already after 5 minutes, if you go back in, you can see that it has spread throughout the entire surface, forming this very thin, elastic hydrogel depot with the API encaptured in there. We have robust preclinical data showing that it's safe and it's feasible. We know that we can load this gel with different therapeutic modalities, and we have streamlined, scalable processes available to really produce this at clinically and commercial scale. So we are now leveraging this technology to create our own broader pipeline of intraperitoneal drug products. We have 2 programs, one on the peritoneal spreading from colorectal cancer and a second program on the ovarian cancer, ranging from the encapsulation of traditional chemotherapeutic drugs, but also addressing the more emerging therapeutic modalities such as DNA repair agents and immune checkpoint inhibitors. We have in-house programs, but also developing together with our strategic partners. And one of the projects that I'd like to share with you to show the potential is on the intraperitoneal delivery of toll-like receptor agonists. It's a very promising class of therapeutics that is nowadays used to locally activate the immune system to actually fight the cancer on site. However, the clinical implementation of such molecules is limited because of the systemic side effects. So you get a cytokine storm that prevents their application. And what we show here is that if you apply these molecules in the form of a gel directly intraperitoneally, that we were able to induce an increased cytokine response at the peritoneal space without this plasma exposure. and that this is actually a very effective event is shown in this animal experiment where we induce these peritoneal cancer in mice, and either by giving these TLR agonists in the form of gel with or without the presence of an additional chemotherapeutic, we were able to actually reduce the tumor growth and even cure these mice. And very impressively, when we re-challenged the surviving animals, they showed to be resistant to reforming any new tumors. So they have really built up a long-term immune memory following this local approach. Then to our lead program, that's UPT211. It's a new formulation of mitomycin C. It's an existing chemotherapeutic that's globally widely accepted as an intraperitoneally cancer agent. We have preclinical data on in 2 species, toxicity and safety, and we are doing the production ready for CTA-enabling studies. What we have shown here is very impressively is that in a very aggressive model of colorectal spread to the peritoneum, that with one single dose without any surgical removal, we were able to cure 78% of these animals. And again, similar to the previous slide, we were able to reduce the systemic exposure here. So these data show that this approach is not only more effective, but it's also more safe to use than the standard systemic products. So with the lead program, we are now entering the IND-enabling phase. We want to go for a clinical trial application filing by the beginning of 2027, entering then a combined phase 1A, phase 1B, early 2027, one with the first clinical data available early 2028. And we have designed this study in such a way that we can continue with a combined phase 2, phase 3 that would position us for conditional approval in 2031. So to give you an overview of the market that we are addressing with this product, this is just the European market of the colorectal cancer patients. We are addressing at least 45,000 patients in Europe, and a conservative estimate indicates that we should be able to obtain more than an NPV of over 450 million euros within the first 5 years after commercial introduction. And to put this in perspective, with this proposition we tap into what we see as a growing interest of pharma in this localized drug delivery, has been illustrated by the recent FDA approval of J&J's Inlexzo and also Susduri from Eurogen, both in the uro-oncology space, where they have expected peak sales of $5 and respectively $1 billion. So the team, all founders are still involved, and we have further expanded it with seasoned entrepreneurs and experts in the field of, and specifically, formulation and drug development in oncology. And very importantly, we are supported by key opinion leaders in this field of peritoneal oncology that is global, but still very small. But they provide us with an excellent position for further clinical evaluation, as we have access to the clinical research centers. So with this, we are currently raising for a Series A to bring the lead compound to the true clinical phase 1, phase B via a CTA approval, and also advance our clinical or our pipeline programs. We have several investors already lined up for this round, but we are interested to speak to others to join them. And of course, I'm also here to speak with partners as our technology is ready for partnering, either on our existing portfolio candidates or on any new assets that one might be interested in to deliver intraperitoneally. So if you're interested, I'm happy to speak to you. Thank you.